The overall goal of this procedure is to isolate Mesangiogenic progenitor cells, or MPC, from the human bone marrow by selective culture conditions for testing both their mesangiogenic and angiogenic potentials.
Mesangiogenic progenitor cells have been identified at low percentage within mesenchymal stromal cell culture optimized for clinical application, when FBS has been replaced by human serum.
This technique is unique in that it allows the consistent isolation of a large number of highly purified MPCs suitable for sprouting angiogenesis and the generation of synchronized MSC cultures.
We discovered this method when, after trypsin digestion of MSC culture in autologous serum, few fried egg-shaped cells remain attached, but differentiated into MCs after FPS treatment.
To isolate human bone marrow mononuclear cells, or MNC, begin by diluting 5 to 10 milliliters of fresh bone marrow up to a total volume of 50 milliliters with DPBS, and mix the cell solution by inversion. Next, equally distribute 25 milliliters of cells into 2 new 50 milliliter conical tubes, and add 25 milliliters of fresh DPBS to each tube for a second mix by inversion. Then rest the cells at room temperature to allow the mineral bone fragments and fat to separate from the solution. After 10 minutes,